Hello everyone. This is the next slide of level 1, chapter 8, part 2. If you like these PPTs, please subscribe and share to other your colleagues. So, types of respiration in plants with glycolysis, Krebs cycle and termination oxidation. Living organisms require energy for all activities like movement, absorption, reproduction, or even breathing. The energy required is obtained from the oxidation of food during respiration. Cellular respiration is the process in which glucose is broken down to release energy. Breaking down of complex molecules takes place in the cytoplasm and in the mitochondria to produce energy. Breaking down of the C-C bond of complex compounds through oxidation within the cells leading to release of energy is termed as respiration. The compounds that get oxidized are called respiratory substrate. Energy released during oxidation is not used directly but utilized in the synthesis of ATP, which is broken down when energy is required. Therefore, ATP is known as the energy currency of cells. Types of respiration in plants. The process of respiration requires oxygen in plants. Oxygen is taken in by lenticels, stomata and root hairs. Plants can get along without respiratory organs because plants don't have great demands for gas exchange. The distance that gases must diffuse in the large plants is not great. During photosynthesis, oxygen is released in leaves and diffuses to other parts of leaves. During the process of respiration, oxygen is utilized and carbon dioxide and water is released along with energy molecules in the form of ATP. Types of respiration: aerobic and anaerobic. Aerobic respiration is of three types: glycolysis, Krebs cycle and terminal oxidation. Aerobic respiration. Aerobic respiration is an enzymatically controlled release of energy in a stepwise catabolic process of complete oxidation of organic food into carbon dioxide and water with oxygen acting as terminal oxidant. Glycolysis. The scheme of glycolysis was given by Embden, Meyerhoff and Paranas. It is also known as the EMP pathway. Glycolysis is the partial oxidation of glucose or similar hexose sugar into two molecules of pyruvic acid through a series of enzyme mediated reactions releasing some ATP and NADH2. It occurs in cytoplasm. In plants, glucose is derived from sucrose or from storage carbohydrate. Sucrose is converted into glucose and fructose by enzyme invertase. Glycolysis starts with phosphorylation of glucose in presence of enzyme hexokinase to form glucose 6-phosphate. One molecule of ATP is used in this process. In next step, glucose 6-phosphate is converted into fructose 6-phosphate catalyzed by enzyme phosphohexose isomerase. Fructose 6-phosphate uses another molecule of ATP to form fructose 1,6-biphosphate in presence of enzyme phosphofructokinase, one for protein and peptones. In glycolysis, two molecules of ATP are consumed during double phosphorylation of glucose to fructose 1,6-biphosphate. Two molecules of NADPH2 are formed at the time of oxidation of glyceraldehyde 3-phosphate to 1,3-diphosphoglycerate. Each NADH is equal to 3 ATP, so that net gain in glycolysis is 8 ATP. Pyruvic acid is the key product of glycolysis. Further breakdown of pyruvic acid depends upon the need of the cell. This is the complete flow chart, like glucose to glucose 6-phosphate, glucose 6 to fructose 6-phosphate, fructose 6 to fructose 1,6 biphosphate, fructose 1,6 by 2 triose phosphate, glyceraldehyde 3 phosphate, then 2 molecules of triose biphosphate, then 2 triose phosphate, 3 phosphoglyceric acid, then 2 phosphoglycerate, after it 2 phosphoenol pyruvate, and last one is 2 pyruvic acid. Pyruvic acid that forms as a product of glycolysis may undergoes three different processes. Lactic acid fermentation. In this type of fermentation, pyruvic acid is converted into lactic acid by the presence of lactic dehydrogenase enzyme. Alcoholic fermentation. In this type of fermentation, alcohol is produced from pyruvic acid. This is glucose, glyceraldehyde, 3-phosphoglyceric acid, phosphenol pyruvic acid, then pyruvic acid, it goes into ethanol and then lactic acid. Krebs cycle. Oxidation of pyruvate to acetyl coenzyme A is done to produce CO2 and NADH. The reaction catalyzed by pyruvic dehydrogenase requires the participation of several coenzymes including NAD+, pyruvic acid plus coenzyme A plus NAD plus magnesium plus acetyl coenzyme A plus CO2 plus NADH plus H plus pyruvic acid. The acetyl coenzyme A enters a cyclic pathway called TCA cycle or Krebs cycle. TCA means tricarboxylic acid cycle. So TCA cycle or Krebs cycle was discovered by Hans Krebs in 1940. This cycle is called TCA cycle because the initial product is citric acid. Acetyl coenzyme A combines with OAA oxaloacetic acid and water to yield citric acid in presence of enzyme citrate synthetase to release coenzyme A. Citrate is then isomerized to isocitrate. It is followed by two successive steps of decarboxylation leading to the formation of alpha-ketoglutarate acid and then succinyl coenzyme A. In the remaining steps, succinyl coenzyme A is oxidized to OAA, allowing the cycle to continue. There are 3 points in the cycle where NAD plus is reduced to NADH2 and 1 point where FAD is reduced to FADH2. A molecule of glucose produced 2 molecule of NADH2, 2 ATP and 2 pyruvate while undergoing glycolysis. The 2 molecules of pyruvate are completely degraded in Krebs cycle to form 2 molecules of ATP, 8 NADH2 and 2 molecules of FADH2. This is the flow chart of Krebs cycle. Start from like citric and citric coenzyme A, citrate, then oxaloacetate, malate, fumarate, succinate, succinyl coenzyme A, oxaloglutarate. Terminal oxidation. Terminal oxidation is the name of oxidation found in aerobic respiration that occurs towards end of catabolic process and involves the passage of both electrons and protons of reduced coenzyme to oxygen to produce water. Electron transport chain. The metabolic pathway through which the electron passes from one carrier to another inside the inner mitochondrial membrane is termed as ETC or mitochondrial respiratory chain. Electrons from NADH produced during the citric acid cycle are oxidized by NADH dehydrogenase and electrons are transferred to ubiquinone located within the inner membrane. Ubiquinone also receives electrons from FADH2 which is transferred to cytochrome C via cytochrome B complex. When the electron pass from one carrier to another via the electron transport chain, they produce ATP from ADP, an inorganic phosphate. The number of ATP molecules synthesized depends upon the electron donor. Oxidation of one molecule of NADH gives rise to 3 molecules of ATP, while oxidation of one molecule of FeD2 produces 2 molecules of ATP. This is the flow chart of ETS, electron transport system. The energy released during ETC is used to make ATP with the help of ATP synthase, which consists of two major parts, F1 and F0. F1 is a peripheral membrane protein complex having site for synthesis of ATP from ADP and inorganic phosphate. F0 is an integral membrane protein that forms a channel for protons. For each ATP produced, 2H plus passes through F0 from the intermembrane species to the matrix down the electrochemical proton gradient. This is the flow chart. Outer side and inner mitochondrial membrane, and in the right side you can see matrix. Points to remember: breaking down process within living organism is called catabolism and the synthesis process is called anabolism process. So respiration is an amphibolic pathway. Respiratory quotient is the ratio of the volume of carbon dioxide produced to the volume of oxygen consumed in respiration over a period of time. RQ is equal to 1 for carbohydrate and less than 1 for proteins. These are the questions arises from this chapter. Why do plants respire? How do plants intake oxygen? Do plants respire at night? Do plants photosynthesize at night? What is ATP? So we will meet tomorrow again. Thank you.